Next up we got nucleic acids, important polymers that are made up of subunits called nucleotides, and they include DNA, RNA, and ATP. There's also DNTPs, ADP, AMP. But for now, DNA, or deoxyribonucleic acid, you better know that by now, was discovered by James Watson, Francis Crick, and Rosalind Franklin, the real one. But sadly, it was named the Watson-Crick model of DNA, despite Rosalind Franklin's work being stolen, pretty much. But yeah. Basically, a unit, nucleotide, consists of three components. The pentose sugar, called deoxyribose, because it's specifically missing an oxygen somewhere. There's a phosphate group, PO4 3-, and the nitrogen base, which can be either a purine or a pyrimidine. Purines are double-ringed, G and A, Ag, pure, like silver. Silver is a pure element, Ag. Pyrimidines are single-ringed, cytosine and thymine. The structure of a DNA nucleotide: there's the PO4 3- group, the phosphate group in the top left, the nitrogen base on the right here on the first carbon, and this sugar base is called a pentose sugar. So it's deoxyribose because on the second sugar here, there's supposed to be another hydroxide here for it to be a regular ribose sugar, but it's missing the oxygen, which is important for stability because oxygen is quite a reactive element. Nucleotides of DNA join together to form a polymer known as, well, the DNA strand, in which the phosphate group on the fifth carbon of one nucleotide links to the third carbon of another through dehydrated synthesis. So what this means is the hydroxide here, and water is removed to create a covalent ester bond here. Or no, they call this a special kind of bond, a phosphodiester bond, because of the way it's linked up and down. So water is removed from the top here and the bottom here, and this is how DNA are linked together through this phosphodiester bond. And water is removed, obviously, so that's a product. And as a result, DNA is a polynucleotide, 3.3 billion chains long around. The sugar and phosphate form the backbone from which the nitrogen bases stick out. So if the phosphates are linking up here, then the nitrogen bases are sticking out like this. There is a 5' end and a 3' end, 5' meaning the end which the phosphate group is on, the fifth carbon, and the 3' end where this carbon is. So this is the 5' and 3' end. And you'll notice the opposing DNA strand, since we know that DNA is double-stranded, has the 5' end and 3' end reverse. This is called the anti-parallel structure. Extremely important word. So DNA consists of two strands held together by hydrogen bonds between the bases. So they're not properly covalently bonded, but there's hydrogen bonding between these two nitrogen bases, and that's how it's held together. So these kind of form the ring, like the rungs on this ladder that you see DNA commonly depicted as. So it's the alpha helix. But remember, alpha helix can refer to a second degree protein. DNA is not a protein, but it does have this structure, the alpha helix structure. So there are triple bonds between cytosine and guanine, and there are double bonds between thymine and adenine. I remember it by saying GCAT, GCAT, GNC, the triple bonds, A and T, the double bonds, in decreasing order. And this is complementary base pairing. If you're describing it in a write-up, I would write complementary base pairing once, in brackets put CBP, and then you can just use CBP inside, because this is a commonly recognized little acronym. So RNA, ribonucleic acid, is similar, but it is using ribose sugar, and all instances of the thymine base is replaced with uracil. So yeah, now uracil is a pyrimidine, and it's the one double bonding. And RNA is single-stranded, is the most important distinction. I've seen questions where they're like, what is usually used to differentiate between DNA and RNA? And the answer is that RNA is single-stranded. So this is the most important fact. So three groups once again: phosphate group, nitrogen base, and to hold all of it together, the pentose sugar. Except now it's a ribose sugar, and there's a extra oxygen to make a hydroxide group on this second carbon. Finally, adenosine triphosphate, nucleotide functioning as an energy carrier in cells. It consists of ribose sugar, adenine, and phosphate groups. So you'll notice it's pretty much the exact same as an RNA nucleotide, except there are 3 phosphate groups. And it exists as a single nucleotide only, meaning that no ATPs will be dehydrate synthesized with each other. So this is how it's depicted, exact same as an RNA nucleotide, except this phosphate group has 3 phosphates here instead of 1, and you must only have the adenine nitrogen base. You'll notice that there are high energy bonds depicted by this squiggly arrow, or squiggly line conjoining them. And when one is broken and used up, it gets transformed to ADP, meaning diphosphate, so there will only be 2 nitrogen bases left. And afterwards, AMP, which if you recall, AMP will just be a RNA molecule of the adenine variant, because it's, chemically, biochemically, exactly the same. So an RNA nucleotide of the adenine type. Okay, DNA replication, very big, lot to explain. DNA replication occurs in the nucleus and is a process where exact copies of double-stranded DNA are produced. Double-stranded DNA is made into new double-stranded DNA in a semi-conservative fashion. So it involves several steps. First, the DNA is unzipped by DNA helicase, like so. There's the 5' end and the 3' end, but it doesn't matter. It is unzipped in the direction of the complex. In the nucleus, many free-floating nucleotides, each with 3 phosphate groups called DNTPs or deoxyribonucleoside triphosphates, complementary base pair, with the exposed unzipped nitrogen bases, since what helicase did was basically tear apart those hydrogen bonds in the middle, so you could add more DNTPs to them. And they'll be added in a 5' to 3' direction on the antisense strand. So if you look here, this one is the antisense strand. Even though it says 3' here, it's because you're adding them in a 5' to 3' direction, and since they'll be anti-parallel even when you synthesize them, then you're adding it to the 3' end first. So 3' down to 5', you're adding in the 5' to 3' direction. So enzyme DNA polymerase 3 adds the DNTPs and joins them, the sugar phosphate backbone, covalently through condensation, so water removal, in the same direction as the replication fork. So alongside helicase in the 5' 3' direction, polymerase 3 is acting at the same time to add these DNTPs, and it will join the sugar phosphate backbone covalently to make sure they're stuck on. So the antisense strand will keep building on and on, but notice how we haven't talked about the sense strand yet, because there is an issue. Since the sense strand is the 5' to 3' strand, you can't add in a 5' to 3'. That wouldn't be anti-parallel anymore. So Okazaki fragments are created instead. So as DNTPs cannot be added in the same direction as the replication fork, replication of sense strand occurs in fragments, known as Okazaki fragments, named after the Japanese couple that discovered them. So the sense strand, there are RNA primase adds a short length of RNA to act as an anchor, as a primer, which allows DNA polymerase 3 to add DNTPs and join the sugar phosphate backbone in a 5' to 3' direction. Like, imagine when you're climbing up a mountain and you set a little hinge into the side of the wall, so you can climb up that short amount, but then you have to set the next hinge on and on and on. That's basically what's happening. That they're setting a little thing and then going back down a little, and then alongside the replication fork, set a little thing and then move back a little. Because DNA can only be replicated in this 5' to 3' direction. Really important. So only short lengths, around 1,000 to 2,000 DNTPs at a time, are formed due to these opposite direction movement. So the short DNA fragments are known as Okazaki fragments. DNA polymerase 1 removes the RNA primer and replaces it with DNA on the sense strand. So polymerase 3 just joins it straight up on the antisense strand, and polymerase 1 removes the anchors and replaces it with DNA. So they both do add DNA, but polymerase 1 removes RNA primer, then adds. As a result, a nick is left behind. Great guy. DNA ligase seals up this nick by making sugar phosphate bonds, so you can think of it like glue. And new double-stranded DNA are termed semi-conservative, because if you think about the way this DNA was made, we're adding DNTPs to the older strand with the newer strand, so it's half old, half new. So your one DNA strand results in 2 strands that are 1/2 old, 1/2 new. And this is done to prevent mutations and to ensure that DNA is replicated the exact same way every time. So protein synthesis. Proteins are essential polymers that, we talked about this earlier in an earlier video. But how are they made? They're made at ribosomes, which require a code from the nucleus. DNA, what we just replicated, contains every single instruction for production of all molecules required by cells. But the DNA is too large to leave through the pores in the nuclear membrane, which means we need to make pre-mRNA and mRNA for it to be able to leave. This intermediate molecule is called messenger RNA, mRNA. And there are 2 steps involved. Transcription, which turns the DNA into mRNA, which happens in the nucleus. Then translation, which turns this new mRNA into the amino acid sequence or the polypeptide. For transcription, DNA unzips with RNA polymerase, not helicase. RNA polymerase does it at the relevant section known as a gene. A gene is a segment of DNA that specifies the amino acid sequence of a protein. So only the relevant area is unzipped, and that's the area that codes for one protein. And the free-floating RNA nucleotides in the nucleus, complementary base pair with these exposed nucleotides that we just opened up with RNA polymerase. On the antisense strand, starting at this promoter region, the letters TAC are the codon for the promoter region. And RNA nucleotides are added starting with TAC on the antisense from a 5' to 3' direction. RNA polymerase will join these sugar phosphate backbones, so RNA polymerase is really carrying transcription here of the newly synthesized RNA nucleotides. And RNA nucleotides keep adding and adding until the terminal region, ATT, ATC, ACT, is encountered on the antisense strand. There's I don't really know how to remember this, but I just remember ACTAT, and then I just rearrange the letters for the last one. This newly formed mRNA leaves the nucleus and heads towards the ribosomes in the cytoplasm. But before that happens, the immediate product of this transcription process, which is what we just did, start at TAC, end at ACT, RNA nucleotides adding in a complementary base pair, which means the opposite one is being added from 5' to 3' direction by RNA polymerase, the carry. The immediate result of this is pre-mRNA, which must be modified through RNA splicing. So introns are removed and exons are kept. I don't know what scientist came up with this, but in means removed, X means kept. So maybe you can think of it as in gets left inside of the nucleus. So introns are removed and exons are kept. And exons are the coding portions. So those are the really important portions to make mature mRNA, which is then sent to the ribosomes. So this starts the translation process. So after they're sent to the out to ribosomes in the cytoplasm, known as polysomes, they're the ones in the bundles, or the ones in the rough ER, which are right next to the nucleus. Then they'll go through the process known as translation. So each amino acid is coded by a group of 3 nitrogen bases called a codon. So all these letters AAU, AAA, the codons are written in mRNA because that's how that's the message we're sending to the ribosomes. So they code for specific amino acids such as asparagine, lysine. AUG is special because if it occurs at the start, if it's either starts, it indicates that this is the start of the codon, the start codon, or it indicates methionine, depending on where it is found in the RNA strand. UAA, UAG, and UGA, which you notice are the complements for ATT, ATC, and ACT, serve as the terminator region, the stop. So there are 20 different amino acids, but each may have more than one codon, and genetic code is termed degenerate for this reason, which means that a few of the codes may still code for the same thing, even if they're mutated. So serine, I'm guessing it's pretty important for it to have so many. UCU, UCC, UCA, UCG, AGU, AGC. Don't remember these. You'll be given a dictionary for codons if they're really needed. This is these are all the variants that serine can be made from. So this is a 5-step process. So I'm going to start explaining now. Initiation. A small ribosomal subunit, which is this little flat thing underneath, binds to the 5' end of the mRNA. And an initiator transfer RNA, known as a tRNA molecule with the anticodon UAC. An anticodon is the opposite to the mRNA. And it's UAC because our start codon is AUG. Complementary base pair, so start codon And the role of tRNA just basically carries amino acids. However, the start one isn't actually an amino acid. It's just a code for start. The large ribosomal subunit binds with the small ribosomal subunit. So a big egg above this little pallet. Initiator tRNA occupies the first binding site, and the next binding site, which is now open, is ready for the next tRNA. And only 2 tRNAs at most can fit on the ribosome at any time. So a peptide bond is formed between the amino acids carried by tRNA. So notice that the next one, GCC, codes for alanine. So the complement CGG, the anticodon, will be finding this GCC. It'll bind here. And a peptide bond will be formed between the acids at the top. Elongation is where the first tRNA is removed and the second one stays. And then a new tRNA is put onto a third slot. So the start codon, for instance, is removed now. And everything shifts over. A peptide bond between the second and third, and this continues until the UGA is found. The stop codon UAA, UAG, and UGA, which once again is the anticodon for the complement for ACT, ATC, ACT. Wait. Oh well. But initiator tRNA is released and it finds its appropriate amino acid. The ribosome translocates, which means that the tRNAs are moving over by one and shifting over until you reach the end. So termination, once this stop codon is encountered, a release factor binds to the UGA site, and the amino acid, or the called a protein or polypeptide, is released along with the mRNA and ribosomal subunits. Termination, so everything just flies off, and it's ready for reuse. There's a little biochem reminder here that dehydration synthesis, this is what's happening, release of water. So there's a lot about mutations, which I'm not too strong on, but mutations are changes in genetic code that are passed on to subsequent cell generations. They may arise spontaneously due to radiation, and they may promote genetic diseases and cause cancer. Chromosomal mutations. So chunks of whole chromosomes are altered. So you need to remember that chromosomes are the largest chunk of DNA. Then I think it goes down to genes. Then it goes down to the strand on its own, like sequences. So a chromosome is a massive chunk of DNA, and prior to any structural change is the first side, and the second side is homologous chromosomes, showing translocation. Pieces of these chromosomes, like these non-homologous chromosomes, which means they don't match up, like this ABCD, ABCD, EFG, HI, they get mixed up. And this is the case for Down syndrome. Chromosome 21 and 14 are mixed up. Deletion is the loss of an entire piece of a chromosome, such as in Cri du chat, where parts of the 5th chromosome are missing. So the F and G segment are missing from this gene. Duplication means that more than one copy of the same gene is present in the chromosome. And this, for example, the gene letter C here is duplicated. Inversion is where the portion of a chromosome breaks loose and rejoins with the ends reversed. So the normal one, the F and G were swapped, and the H and I are swapped and it's flipped over. Just a big mess. Gene mutations, nitrogen base sequences altered rather than, so gene is the smaller segment, the chromosomes are the larger macroscopic view. Gene mutations, we have normal, this is the normal strand, like the cat ate the rat, for example. Obviously these aren't actual bases, this is my teacher being a very smart individual. Deletion, one nitrogen base is missing or deleted, and suddenly everything doesn't make sense because they have to follow in groups of 3 if you remember the codons. So now it's the, atta, tet, her, at, and it's become nonsense. There is no stop codon if the stop codon was rat. At least the start is still intact, but this means you're starting a nonsense code for some protein that doesn't exist. Addition, an extra nitrogen base is added in between, so the stop codon is now displaced by 1, and your start codon will stay the same. But this is still very catastrophic, it's still nonsense. Substitution, one thing was replaced, the rat ate the rat, that doesn't make any sense. It's 1 point mutation. So it might be a one base difference, and I think this is the case with hemoglobin. So sickle cell anemia, anemia, hemophilia. Yeah, sorry. So sickle cell anemia is a change in the 6th amino acid, which causes, I think it changes to valine rather than glutamate, yeah. Which causes the abnormal protein to be formed, and the red blood cells become more crystalline. So victims are out of breath easily because their blood cells can't carry oxygen, and it's quite painful, and it's more common and prevalent among Africans. So glutamate, so it's coded for by GAG, which causes a normal red blood cell, but a substitution of one base only into uracil causes valine, which causes this sickle cell shape. Hemophilia is missing clotting factor. Oh, hemophilia is the missing clotting factor. This is sickle cell anemia, which means that you don't have the factor 8 protein, which we learn about later. But the stop codon occurs too early and cuts off the protein, and makes it so that you don't have the clotting factor. If you get a cut, you will start bleeding out and you'll probably die. G mutations can also occur during meiosis. So mutagen, so meiosis is when they form 4 diploid cells. So mutagen are haploid cells. Mutagen, environmental factor that may increase the risk and chance of mutations. So there's 2 types, chemical mutagens and radiation. So chemical mutagens can happen from food additives such as nitrates and nitrites in meat, hallucinogenic drugs, manufactured chemicals such as DMDM, hydranatoin, and pesticides. Some natural foods contain mutagens and anti-mutagens. So radiation, non-visible short wavelengths of radiation believed to be mutagenic. X-rays, gamma rays, UV rays are all examples of this, so that's why you need your sunscreen. May cause germinal and somatic mutations. Somatic means of the body, germinal means of the children. A carcinogen means something cancer-causing. Cancer cells contain genes called proto-oncogenes that can become oncogenes, and that's just like a little, I don't know, factoid that IB wants you to know, I guess. Recombinant DNA, also known as genetic modification. For example, when a baby lacks growth hormone, this can be solved with genetic modification, recombinant DNA. So what happens is the human gene for growth hormone from someone else maybe is extracted, and the removal of a vector called a plasmid in bacteria such as E. coli. Like how I mentioned earlier, there is a nucleoid region and they have plasmids instead. You can use a restriction enzyme to cleave a portion of the plasmid, and DNA ligase, which was the glue mentioned earlier, is used to anneal the growth hormone gene we removed earlier to this plasmid. Then the plasmid is reintroduced to the E. coli, and it will start dividing and creating this growth hormone as if it was its own. And this is known as recombinant DNA because this DNA is not natural and it's 2 different species of DNA added together. And the bacteria will keep dividing exponentially like this to produce a lot of growth hormone. Insulin was also made in this way. Then there was PCR from the lab that I hope you remember, polymerase chain reaction. And yeah, just to spark your memory, we use Taq polymerase, heat, it's a thermocycler, etc. Alright,